Good afternoon. Let's start with some global statistics. At the end of 2015, there were approximately 36 million people infected with HIV. As you can see in my background, which is showing the global infection rate, Sub-Saharan Africa is the most affected region, with 69% of all people living with HIV. So we need a cure, sooner rather than later. How do we get to that? HIV virus contains proteins on its surface. These proteins can attach to the human cells and fuse the human cells to the virus. Ultimately, infection happens. There is a major protein on the surface of HIV which contributes to HIV infection process that is called GP41. In order to develop a vaccine for such a complex virus like HIV, we need to have a lot of detailed information from the structure to the dynamics to the composition of the system. There has been lots of study about the structure of the protein, but the role of GP41 and how it interacts in the viral membrane is still unclear. That is a missing information that can help HIV vaccine development, and in my research, I would like to find that out. To know more about the structure and behavior of GP41, we need to locally monitor the protein. In my case of a study, I'm using what I call a spy molecule that can be attached to any part of a protein or even the virus cell membrane. The advantage of the spy molecule is that it does not change the structure or biology of the system, and also I can conduct my research in a native environment. So to communicate with the spy molecule, I need to use magnet. The interaction of the magnetic field with the spy molecule provides signals that can be collected using the spectrometer. These signals can contain information about the motion, location, as well as the dynamic of the protein GP41 and its interaction with the viral membrane. We have mimicked several different viral composition and realized that cholesterol has a major role in GP41 interaction with the human cells. Also, we have used a lot of antibodies, and in order to understand how various types of antibodies are interacting with the virus and which one is more potent to block its activity. Hopefully, all this information about the structure and behavior of GP41, as well as its interaction, can be used in future researches to develop a practical vaccine. Thank you for listening.
Thank you.